Hi, this is Alan at Atlantis, and I'm going to talk to you today about a new strain of competent cells we have called Soluble 21. I spend a lot of time expressing proteins in E. coli, and one of the things that I'm constantly faced with is so many of these recombinant proteins are just insoluble. And I'm sure anyone who's ever tried to express proteins in E. coli will find that more than half the time they're going to be insoluble. So I usually try the usual suspects. I'll lower the expression temperature, which will work for less than 10% of these insoluble proteins. Sometimes I'll be a little finicky with the expression construct. I'll move where the purification tag is. I'll add a GST tag, something to improve the solubility of the recombinant protein, and that rarely works. Some of the other things that people commonly try is that they'll actually express the protein in an insoluble form, purify the inclusion body, and then try in vitro to unfold and refold the protein so it does have the solubility that you require. That also rarely works. Usually what we end up doing is we move to other expression systems like yeast or insects or mammalian cells, but all that is a lot of work. So what we've done here at Atlantis is that we've tried to create a version of BL21DE3, which is the most commonly used protein expression strain, and we're trying to create a mutant that has better solubility characteristics than the parent strain. So what we did was we carried out a repetitive mutagenesis and enrichment program on BL21DE3 using test proteins that were completely insoluble. And when we were done with this project, we created a mutant form of BL21DE3, which we call Soluble 21, which now has improved solubility characteristics with a number of proteins. Once we made this strain, we had a panel of 21 completely insoluble mammalian proteins that we put into the new strain. And when we tested each of these 21 proteins, and they were a wide variety of activities, they were repair enzymes, they were kinases, they were phosphatases, they were metabolic intermediate enzymes. What we found was of those 21 proteins, 15 of them were actually now soluble to some degree in the new strain, and they were completely insoluble in the standard BL21DE3. Let me show you some of the data which is present on our website. In these panels, we're showing 5 different mammalian recombinant proteins, and in each case, we have size standards, we have the Soluble 21 strain, and we have the standard BL21 strain. And as you can see in panel number 1, in panel number 2, in panel number 3, then if I scroll down, you can see in panel number 4 and 5, in all of these cases, there is some soluble protein, or in some cases, quite a lot of soluble protein where there wasn't before. Now, in the cases where there's very little, if we need to obtain a substantial amount of the soluble protein, in E. coli, it's very easy to grow larger cultures. And there were, as I said, of our panel of 21, 6 of the insoluble proteins did not express any soluble product, so it clearly does not work for everything, but in our hands, it works for a majority. Now, we find that we see improved solubility in the Soluble 21 strain if we grow our cells in minimal media and we grow them at low temperature. However, a couple of the clones that we have actually perform better at 37 degrees in rich media. So there is some empiricism involved in using the strain to try to create the best conditions for your particular recombinant protein. But in the cases of our group of 21, 15 of them we were able to make them soluble in the new strain. Now, we have available chemically competent Soluble 21, so if you already have a construct, all you need to do is transform it in, and then you can know your results in just a couple of days. If you're in the process of creating a protein expression construct, we have electrocompetent versions of Soluble 21. They have an efficiency of greater than 1 × 10 to the 10th transformants per microgram. And in that case, you could actually create your construct directly in the strain you're going to be analyzing, and you don't have to go through an intermediate strain like DH5 alpha and spend time having to do mini preps and retransform it into the protein expression stream. I hope you found this information useful. Thank you for listening.